Good. All right. Hi and welcome. And thank you for everyone who's joining us on Facebook Live as well. So thanks for joining us together. We're meeting on the lands of the Kaurna people and we'd like to pay our respects to their leaders past, present and emerging. We are now joined with Professor Richard DeAndrea and his team from the University of South Australia and clinician Andy Moore from University of Queensland. So from the moment we met Professor Richard in August last year, we knew he understood us and his vision was in line with ours. We have been in contact over the past 6 months and working together to make a change for these AML kids. We've received several questions over the past month, and so we'll go through these first. And if you have any questions for any of us, including myself, please enter them into the chat button and we'll address them at the end of the session. So let's jump right in. First up, we have a few questions that you've asked about my own personal experience. So this is my story. So the first signs that something was wrong with Charlie is he had a lesion in his right eye and he had viruses on top of one another from around 12 months and he was a spewy baby. A few months later, after investigations, Charlie ended up in hospital with pneumonia, and later that night we found out that he had AML, Acute Myeloid Leukaemia. So how long was Charlie's treatment? Charlie's treatment was 18 months with 6 months of intensive chemo and then a 5-month break where we thought we were the lucky ones, that we'd only had 6 months of treatment and that we had gone back to normal life. Unfortunately, after those 5 months, Charlie relapsed and relapsed hard, and the rest of his treatment was 7 months until he passed away. During Charlie's treatment, when did we know that we were fighting a losing battle, and how did we cope with this? Christmas 2020, when we found out that Charlie had relapsed after his transplant, we just knew that it was just too soon after and that the prognosis wasn't good. I Googled and I realized that we had less than 6 months with him and that would be our last Christmas. How we got through that? Hope. Hope is the only way to get through that. And with our family, our friends, and a lot of coffee. When was it that I decided to start Charlie's Rainbow, and why? Only a few months after losing Charlie, I decided that I wanted to make a difference. I wanted to make a change for these kids and no one should have to go through what we went through. I did a lot of research and looked into a lot of charities to find out who was researching paediatric AML, and a lot of them were adult leukemias and not children, or they were going to other things. So I decided to make a difference. And I stumbled across UniSA and their paediatric research. So here we are, Charlie's Rainbow. How is it that I still function, let alone run Charlie's Rainbow after losing my little boy? Well, that is simple, and that is Harvey, Charlie's big brother. He is the only reason that my husband and I get up every morning and put on our big kid boots and get out of bed. We want his life to be as normal as possible, but how can your life be normal when you're missing a part of your heart? There's a poem that I read about a stone in your pocket, and this is what it's like going through the grief of losing a child or losing anybody. So having that stone in your pocket, you walk around all day, some days on a certain angle, it rubs on you and it drops you and you are in pain. Other days you don't notice it quite so much, and this is how it feels day in, day out through our journey. All we can do is stay strong and make a difference. So what do I do to help survive my grief? I do Charlie's Rainbow. And this is my way of spending time with Charlie every single day. I work out, and I work out hard. If I work out, it hurts less than losing my child. And I talk. I talk about Charlie, and I talk about Charlie's rainbow. I talk about the difference that we're going to make, and I spread the word as much as I can. What do we do as a family to keep Charlie's memory alive? Do we have anything that reminds us of him? Well, for those who know me, know that it's all about rainbows and unicorns. When we were in Melbourne, we had to leave Harvey behind in Adelaide because of the COVID logistics. And so we would FaceTime every day. And when we would do this, Charlie would be the rainbow unicorn every single time. He was always spewing out rainbows out of his mouth. So for Christmas, I bought the boys unicorn rainbow onesies, and Charlie was the first. He put his on and didn't want to take it off. So from then on, Charlie has been our rainbow unicorn. Charlie has an older brother, which I mentioned previously, Harvey. And how is he doing? Harvey is amazing. He is resilient, and he talks about Charlie. He talks about the fact that Charlie died, and he tells all his friends about it. And all his friends, when they see a rainbow, their parents message us and say, we saw Charlie today. So to know that Charlie is still making a difference in everybody's lives and everyone is getting to know our Charlie boy just makes a huge difference. So, as I mentioned earlier, we're very lucky to have a group of AML experts here today to answer some of your questions. So Richard, I'm going to put it over to you. Would you like to introduce yourself and your team and tell us about your AML research?
Yeah, thanks, Kelly. That's great for you to go through that story. I'm sure that's really helpful for everybody. So, yeah, I'm at the Centre for Cancer Biology at University of South Australia. I run the Acute Leukaemia laboratory. So we have projects looking at predominantly acute myeloid leukemia and some other related diseases. The team, I'll let Andy introduce himself, I think, at the end. I'll introduce the team here from UniSA. So Saumya, who is sitting on my right, where she's on the screen. She's a mid-career researcher, so she is leading the childhood AML work in the lab. She's got excellent training in genetics and has been working on the mechanisms of disease in AML for quite a while now. And she led the publication that has recently come out, which we'll talk about as we move on. Debora is a senior postdoc in my lab. So she's got extensive experience in leukemia biology as well through some of her previous appointments, and worked with one of the leaders in the international leaders in the field in CML, and has published on CML, AML, and myeloproliferative diseases. And Sheree is a senior lecturer in laboratory medicine at UniSA and does quite a bit of teaching, but also contributes a lot to our research in the lab, and did a PhD in 2017 and has a lot of expertise, particularly in diagnostics, which has been really useful for us to have that expertise in the group as well. And I'll hand over to Andy just to introduce himself. I think it's probably the best way to do that.
Thanks, Richard, and thanks, Kelly, for putting on the event today. I think it's a great opportunity, and we're grateful to work that you're doing to share your story and raise awareness and some fundraising. So Andy Moore is my name. I'm a paediatric oncologist here in Brisbane at the Queensland Children's Hospital. I'm also director of the Queensland Children's Tumour Bank, which is a nationally accessible tumour bank. We collect samples from children with all forms of cancer, particularly AML. I lead Australia and New Zealand's involvement in a large international clinical trial for AML, and part of that, all the samples pass through our lab as part of that trial. So that's a rich resource as well for research, which we make available to expert groups like Richard's lab in Adelaide. So that's where I fit into the scene, and AML is an area of my particular clinical interest.
All right, so Richard, can you tell me what your AML research is about? I know you kind of briefly went into it, but why is this work important?
Yep. So we focus on understanding what causes AML in adults and children, and our focus, we work with people like Andy because we want to make sure our work is geared towards improving the treatment outcomes for patients. Our childhood AML work, we've been using sophisticated genomics technology, which allows us to look at all of the changes, all of the mutations that occur in samples of AML. So this uses some samples from Queensland that Andy talked about, and also a big sample set we were able to get hold of from the US. And we work with experts in cancer genetics here in South Australia, and also people at through the Zero Childhood Cancer Program have experts in childhood cancer there. We work with them to look at all of this data and try and understand the causes of AML from that. And also we have a particular interest in looking at whether there's an underlying risk or predisposition in some of these AML cases that we can pick up through the analysis of these mutations. We also look at, we're also looking again with Andy at ways of enhancing the immune system's ability to kill the AML cells off, and that's a new project. Sheree's leading that in our lab at the moment. So, and we all believe that immunotherapy is going to be a major player going forward with AML. The reason the work's important is because getting back to the cancer predisposition in particular, understanding predisposition in cancer is really important because it gives you clues as to how the cancer is initiated for starters. So from a biology point of view, it's really interesting, and that's certainly one of the reasons we look at it. But it's also critical in AML because, as Kelly pointed to, AML patients can be transplanted, can receive a blood stem cell transplant, which is potentially curative in some cases. And that involves the patient receiving a transplantation of donor blood stem cells, which often come from a family member. So it's critical, obviously, when you're transplanting a patient with a family member's stem cells to understand whether there's any predisposition associated with that, because obviously you don't want the patient to be transplanted and again have the risk again of getting AML. So we need to understand that predisposition. It's a complicated genetic thing to understand. The other thing also is that you can have rare mutations in families that can influence the patient's response to chemotherapy. So some mutations that can lead to sensitivity to the chemotherapy treatment. So that you need to understand that and then pass that information on to the clinician as well. So, and things have changed dramatically with cancer diagnosis now. There's some fantastic new technologies for analysing tumour samples that give you massive amounts of information, genetic information, that all of these factors I've just talked about can be looked at. And it's really important that we have the ways to analyse that data, and we have the mechanisms in place to feed the useful information back to clinicians like Andy so that they can decide about the transplant, whether the induction treatment needs to be changed, whether the donor sample is there any risks associated with that donor sample. So that's something we're working on extensively with Andy and also with the team in Sydney at the Zero Childhood Cancer Program. And that's exciting because all AML patients from mid-year this year will receive full genomics characterization for their AML. So that it's important that we work through that process and how to analyse all of that information and work with the team there to get that information to the clinicians.
So what is groundbreaking and new in these research?
Yeah, sure. We know, I guess I'll let Saumya talk a little bit about this because we've just published some of this work in hematology, one of the leading hematology journals called Blood. So I think Saumya was the lead author on that. So I'll hand over to Saumya. She can talk a little bit about that work.
Thanks very much. So yeah, I was the lead author on a recent study that we've published, and we showed that up to 19% of childhood AML patients have certain rare genetic changes that occur, which can indicate a familial cancer predisposition or a sensitivity to chemotherapy as well. And sometimes these changes may previously be unknown to the family and to the clinicians. And the new technologies that are available now means that we can identify these changes at an early stage, so at diagnosis. And knowing these kind of changes early on is really important, as Richard mentioned previously, particularly to clinicians in terms of weighing up their transplant donor choices and also deciding what kinds of treatments are appropriate for the patients. So that's really what we've come up with in the last year or so. So what are the next steps? What are the steps in this research?
We're now working with, we're having sessions and workshops with the team at Zero Childhood Cancer now to work out the best way to process all of that data that will come through, look through the data for indications of familial predisposition. As you can imagine, there's a massive amount of data that has to be analysed. There's a lot of expertise required to go through that and make decisions on which mutations are in the sample that need to be reported back to the clinician, and obviously there needs to be some discussion between the clinician and the curation team to understand that and make the decisions about the transplant. And there can also be implications, obviously, for genetic counselling. That's the other thing, whether families are referred for genetic counselling. So that's where that project is at the moment. I guess that's the sort of translational phase of that work. We need to really work that through so that that sort of understanding moves into the clinic and the clinicians are getting the benefit of that information. Andy might like to comment on some of that stuff as well, obviously being on the front line with all of this.
Yeah, thanks, Richard. I think the challenge in terms of the next steps, it's actually part of it is a challenge in terms of getting a deeper understanding of what some of these rare events that appear to be predisposing, what they mean in terms of what we call penetrance and how likely they are to cause AML, how big an influence they are in terms of things like donor selection. So all of that is still to be worked through on a number of levels. And that's part of, I suppose, the exciting time in AML research, as it is for other forms of cancer research, in that we have an exponential increase in our understanding of the biology and the abnormalities that have gone on inside leukemia cells, as well as in the germline, and the challenges kind of making sense of all of that, and hopefully helping to inform how we treat patients.
Thank you so much for answering these questions. So today we thought it would be a great opportunity to get together and open it up for you all to ask about AML. So here are some of the questions that we've been asked by the public already. So is there a genetic link in AML?
Well, yes, there is. I mean, AML, like many cancers, is a genetic disease, and it arises from mutations that cells acquire. In the case of childhood AML, these mutations are acquired as the blood system develops in the embryo, and some of the cells acquire mutations as a process during that process, and it's basically down to chance whether one of those mutations is an initiating event for AML. So certainly AML is driven by genetic changes in that developing blood system in the case of childhood AML. And we also, there's a strong genetic links with some genetic syndromes. I mean, there are syndromes like Fanconi anemia, for example, which are rare recessive syndromes that is at bone marrow, it causes bone marrow failure, but in AML, it causes also a very high risk of development of AML in early life. So and there are other genetic syndromes as well that increase risk of AML. So there's certainly a genetic link there. And from the work we've done recently, we know that in some cases we can pick up mutations that may be familial in nature and can be associated with an increased risk of AML. That's not to say every patient that has that mutation will develop AML, like Andy's saying. It's one of those things you have to work through, but certainly we have found mutations that we know are linked to AML development in families, and potentially families aren't aware of that. So there's a lot to be done there, but certainly there is a genetic link. We know that because of some of the genetic syndromes that are very strongly linked to a high risk of AML.
So in saying that, this kind of goes on from that, AML does have genetic mutations like other cancers?
Yep, that's the cause of AML, like many other cancers, is that the cells, the blood stem cells, the cells that normally drive your development of your blood and immune system, they acquire mutations as a part of their normal process. Part of normal life, cells acquire mutations, and in the case of blood stem cells, they can acquire a mutation that causes them to basically misbehave. They become a rogue stem cell. Instead of producing normal blood and immune cells, they produce these blast cells that don't perform normal blood stem cell function. Instead they just keep producing blast cells and proliferate abnormally and eventually overrun the bone marrow and cause the symptoms we see with AML. So yeah, it's caused by mutations in blood stem cells.
So what would be the early signs that we would look for?
Oh, again, I flagged this one for Andy to talk about some of the signs that you see with AML and also some of the recent developments for treatment of AML. I thought Andy's best to talk about that.
Yeah, sure. So a really common question. I mean, we see children who present with childhood leukemia really present with pretty common themes and in many ways similar to common viral infections. Stories that we hear again and again from new patients and their families who present is that everyone in their household might have had the flu or the cold, and everyone kind of got better except the child who obviously had leukemia underlying it, or they seem to get one thing after another, or a grandparent visited and noticed that they all of a sudden looked more pale, whereas you don't tend to notice that from a day-to-day basis. So the symptoms arise as a consequence of having the bone marrow sort of overtaken by the blast cells and the leukemia at the expense of the production of the normal cells. And so you get to a tipping point where the production of the normal red blood cells that carry oxygen around and give us energy eventually is low enough that you start to see some symptoms of tiredness, or if you have a low white blood cell count, for example, you might have fevers, or the leukemia itself can produce chemicals that can stimulate a fever, for example, or bruising due to low platelet count. Again, when that normal bone marrow that's normally producing platelets, when that's overrun enough, when the factory starts to get shut down by the abnormal leukemia cells, then you might notice some bruises that they're not normal, rough and tumble bumps and bruises. And so eventually, you get a blood test and it shows, a pretty abnormal picture and suggestive of leukemia. And again, I tend to counsel families that we don't really see a late or an early presentation in leukemia. Blood goes everywhere, so it's not like breast cancer or bowel cancer that might have spread. The leukemia in the bloodstream sort of goes everywhere, and often children have lots of viral infections, lots of aches and pains and fevers and illnesses during their childhood, and they certainly don't need a blood test for all of those. And the average GP, for example, for thousands of children that have lots of viruses and coughs and colds, they might be lucky or unlucky rather if they see one child with leukemia in the course of their career. So we don't want all these children having a blood test every time they have a fever or a viral infection. But clearly, eventually something's not quite right with the patient that's got leukemia underlying it all, and someone, eventually, let's get a blood test, or they get taken to the emergency department because they're unwell, and they get a blood test, and that's where the diagnosis comes from. So it's not really a case of sort of looking out for symptoms or signs. Certainly, we know in patients where there is a really clear predisposition risk, like as Richard mentioned, Fanconi anemia, where if you've got an established diagnosis, an established predisposition that is known, there's a strong family history, everyone's onto it, Obviously, the threshold for going and getting a blood test for the patient of concern is going to be a little bit lower. Some of those patients might be having surveillance bloods, but again, that's something that needs an individualised consideration. But yeah, again, it tends not to be, for the overwhelming majority of patients, it's not something that tends to be early or late, if that makes sense.
Thanks, Andy. So back asking you again, Andy, have there been any recent developments in the treatment of AML in children in forms of different medications and things?
Yeah, so great question. So the current clinical trial that we have running in Australia and New Zealand, and we do this in collaboration with our colleagues in the United Kingdom and France and Ireland, Switzerland, is exploring how best to give a novel chemotherapy medicine, a medicine called gemtuzumab, which is has a drug attached to an antibody that recognises a particular marker on the surface of AML cells, a marker called CD33. And so that trial is looking at how best to give that new chemotherapy medicine, either in one dose or in 3 small doses. And it's also looking at how best to give the 3rd and 4th cycles of chemotherapy, which combination of medicines to use for those cycles for patients that have what we call standard risk disease. And additional drugs like venetoclax is another medicine that we're opening a study in patients with relapse disease later this year, we hope. And that's a medicine that originated from labs in Melbourne many years ago. The drug development process is a very long time. Venetoclax is a medicine that helps switch on the mechanisms inside cells for programmed cell death. And so cells have normal cells and leukemia cells and cancer cells have either very good mechanisms to either fix themselves if they're damaged or recognize too much damage gone on here. I'm going to, take myself out and shrivel up. And so venetoclax is a medicine that helps switch on, help cells accelerate that cell death pathway in response to chemotherapy. And that we hope, one, improves the response to conventional chemotherapy. And it's also hoped that we might be able to dial down the dose of certain chemotherapy medicines to reduce side effects when we combine it with medicines like venetoclax. There's a few other potential new drugs coming online as well. The menin inhibitors, for example, are a class of drug that we're excited about for patients that have a specific rearrangement of a gene called MLL or KMT2A. Again, it's probably around 30%, 20 to 30% of children with AML will have an abnormality inside their leukemia cells involving that gene that these inhibitors seem to target well. The trials for those are a little way away at the moment, but we're certainly looking forward to when those trials open. Again, it's one of those things that there's always some new developments coming out of the lab in terms of someone identifies a new marker and a new drug or potential new drug, but there's a lot of hurdles and hoops to jump through to get those drugs to be effective in the clinic. But certainly there's a few exciting developments that we're looking forward to seeing translate in clinical trials and getting to patients.
It's so great to see that venetoclax and gemtuzumab are ones that can be accessed a little easier. Charlie had both of these. He did have the MLL gene. So we were able to gain them on compassionate grounds. So, to be able to not have to wait, we had to wait 6 weeks for gemtuzumab. For venetoclax. So, to be able to get onto that, quicker and things like that, as these trials go on is just brilliant. So thank you. That's amazing. So why is AML such an aggressive cancer in children with high rates of treatment failure, relapse, and less than 1 year remissions?
Yeah, look, it's a really good question. I think the thing to point out is the overwhelmingly, the majority of patients will do well. It is with very aggressive treatment, and as you well know, roughly about 70% of patients, which is way too short of 100, will be cured with upfront therapy. Often that may involve a transplant for those who have a particularly high risk of their leukemia coming back. But the disease, it's the relapse rate and that really makes it the aggressive disease that it is. The majority of patients will get into remission. The challenge is keeping them into remission and keeping them at a what we call a deep enough remission. So what we call MRD, or minimal residual disease, MRD negative. So a really deep state of remission, hopefully with the leukemia initiating cells having been taken out by the chemotherapy or their transplant, if that's what they've had. But that's really what makes it an aggressive and difficult disease to treat, is that it has that high relapse rate. And, at best around sort of 40% of patients where that leukemia comes back, if they get to a transplant, it can be cured. But there's still clearly far too many patients who we're not curing with our current approaches, which are pretty intensive. I often describe AML therapy as being certainly not straightforward, but it's straightforward insofar as it's either intensive or really intensive with a transplant, which isn't particularly sophisticated. The supportive care and everything we do is definitely sophisticated. But the general approach with the chemotherapy medicines that we use and the transplant approaches, it's relatively straightforward, and we need to improve that with better drugs, more effective treatments that allow us to get our patients into remission, keep them there without such a reliance on really intensive chemotherapies.
Thank you. So are there many types of AML? Would you be able to explain them and their risk factors?
Yeah, so there's lots. There's lots. I think one of the
threw you under a bus there. Sorry, that one's full.
Yeah, one of the great things of recent years, probably the last 10 years, I suppose, has been our increased understanding of the different subtypes of leukemia. And that's not just AML, but all forms of childhood leukemia and adult leukemia as well, for that matter. But when it comes to childhood AML, we know that it's a very different disease biologically in many ways to adults. Adult disease tends to have occurred over time with a series of random events in sort of blood stem cells that have tipped them over the balance to become leukemia cells. In childhood AML, there's a much higher incidence of what we call gene fusions, whereby genes have got muddled up and mixed around as normal cells have divided, and then on top of that there's additional random mutations and events that help tip it over the balance to become a leukemia cell. And we know that those abnormalities inside the leukemia cell, we've known for quite some time that there's a group of abnormalities that are associated with a lower risk of the leukemia coming back. And we tend to call those standard risk or good risk. And then there are a bunch of abnormalities where the leukemia has a much higher rate of coming back and a much higher risk of relapse. And so we call those high risk or poor risk leukemias. And then in AML, we've tended to bunch all the rest of it in the middle, in intermediate risk, because we just don't have the data to know, is that high risk? Is that benefited from a first remission transplant? Or there are some patients that seem to do okay with chemotherapy alone and they don't need a transplant. We know that when you relapse, if you haven't had a transplant, you tend to have a better prognosis than if your disease has come back after having had a transplant. And so with a deeper understanding of the different molecular subtypes, we've got, unfortunately, we've still got those three groups of patients. We've still got the good risk, the intermediate risk, and the high risk. But what we know now is that we've got a better understanding and also more sophisticated ways through genomic analyses and also other diagnostic techniques in the pathology lab to identify those rare, hard to identify fusions and other abnormalities that we might not have identified 10 years ago, but now we can say, yes, you've got this fusion, that's rare, we know that's bad, you're high risk and you're going to need a first remission transplant. The challenge is improving our therapy, because we know whilst we put you down a high risk path and take you to a first remission transplant, that doesn't always cure those patients. And so it's really a case of a transplant up front, or what we call a first remission transplant, is kind of the most intensive thing that we can do. And so if we know that you've got an abnormality that has a very high risk of relapse, then we take you down that transplant pathway up front. So that's and that creates its own challenges, because whilst we've got a deeper understanding of the subtypes of AML, they're all relatively rare. So we see abnormalities that, some of these gene fusions in AML that are associated with a poor prognosis, they might account for 2% of all AML. And so Australia and New Zealand, we get just under 50 patients a year. So in the course of 2 years, Australia and New Zealand will see one child with this one particular abnormality. And so that's essentially impossible to run a clinical trial to try and find out, is there a new targeted drug specifically targeting that abnormality? Is that going to benefit children with that abnormality? You need to do that on an international scale, very large studies. And even then, you might only see during the course of these trials that might even last 8 or 10 years, you might still only be talking about 10 or 20 children with that specific abnormality. So if you've got a highly specific drug that's targeting that abnormality, you need to make sure, have very good evidence that that's actually going to make a big difference by adding that in. Certainly the holy grail is to add it in and take away some of the other more blunderbuss chemotherapy, but it creates exciting opportunities on the one hand, in that we understand the biology and hopefully we can identify therapeutic targets, but it introduces additional challenges in how we answer the questions and how we introduce new therapies moving forward that are going to be effective and hopefully curative.
No worries. And so I'm just going to skip to one of the questions that someone has asked today, is what is AML compared to ALL? I guess because you're speaking about, the different subtypes of AML there as well, but I just thought, does ALL kind of work in the same kind of way as that?
In terms of the general biology, yes. In terms of, we know that childhood ALL, the way it develops is a generally similar process, in terms of a random abnormality inside a normal cell that was destined to become a normal blood cell, a random event. Sometimes those initial gene fusions occur during embryonic development and are there at birth. We know, for example, one of the common fusions inside childhood ALL is present on studies that have been done on the heel prick blood test cards for when every baby has when they're born. We know that sometimes a gene fusion that we see in childhood ALL is actually present in 1 in 100 babies, but definitely a much lower incidence than 1 in 100 that then go on to develop leukemia. And there's certainly no way of preventing that leukemia, but we know that it's a very, very small percentage of patients that go on to develop leukemia. The point being is that those abnormalities arise in a random kind of way, and what our understanding from the biology, and Richard can speak to this more, is that we've got a pretty good picture of what's gone wrong and how things have gone wrong. What the next steps we need to understand for all leukemias is what happened when, what made it happen, and then of course what can we do to stop it developing its next abnormality and then tipping it over the edge? The childhood ALL is much more common. We would see here in Queensland, in northern New South Wales, we'd see about one patient each week, a new patient. And so it's the most common form of childhood leukemia. It has a much higher cure rate, is much more sensitive to chemotherapy and steroids and other medicines as well. So collectively, close to 90% of children with ALL will be cured with chemotherapy, pretty complex chemotherapy. And again, whilst not as intensive as AML therapy, very complex chemotherapy for ALL, still associated with complications and the need for supportive care. And ALL has this period of what we call maintenance chemotherapy, which lasts just under 2 years. So it's a different lot of intensity and hard work.
Absolutely. We've had, obviously being oncology families, we've had a lot of conversations with other parents about this and seeing the difference between those as well. It's it's hugely different. So what does your vision of a new treatment look like?
My vision of a new treatment is that it's oral. So it's a tablet or a liquid, it's a medicine, and it doesn't put you in hospital. Doesn't make you sick. It targets the leukemia cell, and the leukemia goes away. It's difficult to see that anytime soon. I think there are rare examples of there's a specific switch that's been turned on the leukemia cell, and when you turn it off with this particular drug, it all just goes away. We see that in CML, in chronic myeloid leukemia in adults with medicines like imatinib or dasatinib. Again, and that's in a particular subtype of CML, and that's in some ways it's a very different disease to AML. I think that has introduced that's a kind of a model, and that's the holy grail for all cancers, is that here's the abnormality, here's the switch, here's the medicine to switch it off. That's a challenge. I think for AML, what we will see in the next 1 or 2 decades is better immunotherapies. So we know that there are some really impressive results with CAR T cells in ALL and other immunotherapies. There are some immunotherapies coming online for AML. The challenge is the markers on the surface of the leukemia cell that we target even in ALL, they're also expressed on normal cells. And so even though we have targeted immunotherapies, there are a subset of normal cells that are also taken out. In AML, that's more of a problem than it is in ALL, because the targets that are easy to find and go after in AML tend to be expressed on normal cells like neutrophils. And as any cancer parent knows, neutrophils are good things. And we need them. So
things we can leave the house.
Correct. So the immunotherapies are exciting, and the work that Richard's lab is starting to explore as well. That's exciting and an important area of research that we need to progress. And so hopefully that's where we see some real developments in the next 10 years, because we certainly know that there are far too many patients, particularly with relapsed AML, where pretty intensive chemotherapy is really failing those children and doesn't work. They don't respond, and it's devastating. And we need a better way of doing that, and hopefully immunotherapy approaches will provide that treatment option.
So why are we finding that paediatric research is underfunded? What is it that childhood cancers, why is it that childhood cancers are studied much less than adult cancers? And how can we change this?
Oh, I mean, I can start that off. I think it's obviously childhood AML is a very rare childhood cancer, even amongst the childhood cancers. 3%, I think, of all childhood cancers. So it's a very rare disease, and the common adult cancers obviously affect large numbers of people. Everybody knows somebody that's been impacted by some of the major solid cancers, breast, prostate, colon cancer, and they're a bigger have a bigger impact on the health system. So I think rare childhood cancers just suffer from that fact that they're rare. There has been some good funding for childhood cancer along the lines of the Zero Childhood Cancer Program, and they've run some large studies across all childhood cancers. But the issue with that is that even across all childhood cancers, as I said before, in a large study of 100 samples, there's not many childhood AMLs in amongst that. So even with large childhood cancer studies going on, there's still sparse data coming out on AML. So the challenge with a rare childhood cancer is really to get a study going that's focused on that. And like Andy said, it has to be really an international study. It has to really be across multiple centres and at that sort of level to really generate the data that we need to look across a range of AML subtypes. Because like we said before, it's a complex disease. There's a lot of subtypes. And even if you ran 100 childhood AML samples, some of those subtypes would be represented by only a few samples. So it's just the challenge of it being such a rare cancer and being a heterogeneous disease. So that makes it very challenging. And I think we need to get across to the funding bodies and to the local and federal governments that even though it's a rare disease, the impact is still large, has large impact on families, and we really need to be getting better access to funding for studies and better access to trial drugs, and making sure that the drugs that are being trialled are available in Australia, and working through that using the new scheme that's available. The MRFF is a fund set up by the government. There may be some opportunities through that. Andy and I have sort of been looking at that. But I think that it's challenging because it's a rare, complex disease, basically a rare, complex childhood cancer, even amongst the childhood cancers. Do you want to comment on that too, Andy?
Yeah, you're absolutely right. I think from a trial perspective, there are additional challenges as well, and we need funding for those studies. Running a clinical trial costs millions of dollars. Our current AML trial, thankfully, was supported to the tune of $1 million, and that $1 million goes to a lot of that goes on sample processing and shipping, and the data collection that we need. Obviously, you want to learn things out of a clinical trial. You want to learn and make sure that the interventions that you're doing in the clinical trial, in the MyChild trial, it's 1 versus 3 doses of the gemtuzumab, for example. So you need to have the staff that are collecting all of the data and doing all of the assessments and reporting and all the rest of it. Those staff need to be paid. So everything, as I often say, everything we do costs money. Whether it's in the laboratory with the pipette tips or the tubes or the liquid nitrogen and the electricity for the freezers, the staff need to be paid. Or whether it's a clinical trial, the samples need to be shipped. There's medicines that need to be purchased. There's medicines that need to be shipped from the US or Europe to Australia. We send samples internationally around Australia as well. Everything comes with a price tag, and that very quickly adds up to a lot of money. And so if regardless of whether it's basic science research, translational research, clinical research, it all comes with a cost, so it's a major challenge.
So I guess this is, for either of you to answer, and I'll kind of put the last few together before we go to taking some of the other questions. I think this is what every parent puts pressure on themselves, and they, I guess, want to ask, but is there something that we did that our child got leukemia, or were we exposed to something, or was it an illness that we had while we were pregnant, or or is it something that we could have been exposed to as a child? I went to school with 7 other students over a space of 6 years, and each of us have had a child who's had leukemia of different types. I know 2 of them were AML, and both of those passed away. So is there anything, like, I guess we're learning all of this at the moment, aren't you, where it's sort of coming from, but is there something to say that any of this is connected?
Look, AML is not associated. There are some risk factors for AML, but having said that, a lot of AML patients, there is no known risk factor. So it's not like lung cancer or some other cancers where there's liver cancer where there's obvious risk factors. And I'm sure Andy will vouch for that. A lot of patients turn up, there's no obvious background cause or that they can see. I mean, sometimes there's a family history, and where a really strong family history is pretty rare, and that's sort of Fanconi anemia and that sort of thing. There's obviously a family history, but in a lot of cases, I think even that's pretty hard to pick up. So I think, you're being pretty hard on yourself if in childhood AML to say that it's something that you did. Obviously, as we get more genetic information, there will be more that we understand about the sort of risk in families and whether there is a sort of a slightly increased risk in a particular family due to a particular uncommon mutation or something. But no, childhood AML is not something that's associated with obvious risk family. I mean, there's things like large levels of alcohol consumption during pregnancy and things like that have been certainly tossed up as risk factors, but a lot of those studies, even then the risk associated with that is still to be well determined. So there are some risk factors, but there's not major risk factors like some of the solid cancers where we know of obvious risk factors.
Yeah, again, and that's a really common question, and a question that parents will ask me at a point of diagnosis. And again, it's there's nothing that the parents have done or haven't done or eaten or haven't eaten or whatever. As Richard indicated, we know that there are clearly there are some events, high levels of ionizing radiation, for example. We know that there was a significant increase in leukemia diagnoses after things like Hiroshima. I mean, that's clearly a really extraordinary event. But you would need, significant, if you had multiple CT scans, for example, when you were pregnant and not realized you're pregnant, that would be, if a parent mentioned that in the history, then you'd be thinking, oh gosh, there probably was the fact that you had multiple scans, multiple significant exposures. It's always extraordinarily difficult to tease apart cancer clusters. You mentioned that the school issue, again, that's been looked at from time to time. Our public health units are actually pretty good at identifying when something like that is occurring. We had that, relatively recently in the ABC radio stations here in Brisbane with a number of the staff getting breast cancer, for example. And it's just really hard to tease apart. And again, I think it's in terms of what we then do about that, I think it's kind of, in many ways, it's almost like a car accident is just often being in the wrong place at the wrong time, at the wrong intersection, wearing a seatbelt, weren't speeding, all the rest of it. It's just one of those random, awful life events that's happened.
It's a really bad lottery.
In the case of the leukemia, the wrong blood stem cell was doing, at the wrong point of its cell cycle at the wrong time that it got exposed to whether that was a viral infection and it was trying to do its normal job, or whether it was some other insult that we don't quite understand, or it was a random event that just happened to be the right random event at the wrong time for that particular cell that just put it down a destiny at a cellular level to then make it a bit unstable and develop a second abnormality and then a third that tips it over the edge to become a leukemia cell. So it's one of those things, again, every parent asks and is, why has this happened to us? And
Yeah,
you know, we see that in other elements outside. One of the I think the things when we have to go down to intensive care or other wards, you see patients in any hospital where just, you know, awful chance things have happened to people for no rhyme or reason. And in the vast majority of cases of childhood leukemia, that's the scenario, is that there's no rhyme or reason why this has occurred. But it's certainly a common question, that's for sure, and totally understandable.
Yeah. And even with clusters, you know, you really need detailed epidemiological investigation of a cluster to rule out that it's not due to chance. I mean, unfortunately, you know, sometimes bad luck happens in runs, and you know, it can be a chance event to have sort of a number of cases like that over a period of time in the same sort of region. And, you know, the experts need to look at it and decide, is that a chance event, or is it actually really starting to get outside the realms of chance and should be looked at more closely, and the risk exposures, etc., looked at. But you know, often those things turn out to be just basically a run of bad luck by some people in the same place.
I've just got a couple more questions from those who are watching on today. So are the current best treatments for AML available in Australia, or are these all clinical trials and treatments only available overseas? I know when we were at our wit's end after transplant hadn't worked, but we were only 50 days post-transplant, we were looking overseas, and we were going to end up in St. Louis or somewhere around there as well, or China. That was kind of our options. And so, yeah, are these available in Australia, or you still need to go overseas for the best possible treatment? You're muted.
The trials that we have at the moment, the upfront trial, as I said, that's open in the UK, France, Switzerland, Australia, New Zealand. Similarly, for other childhood cancers and leukemias, we're part of major international collaborations and cooperative groups. So if you walk in the door with childhood cancer in Australia, your treatment will be exactly the same as what it would be in a big centre in the United States. And I think the other thing, just on that geographic sort of difference, a number of centres in Australia are as big or bigger than any of the centres in the United States. And so that's just something that I find myself reassuring some parents as well, in that actually you wouldn't be better if you were in, you know, some famous city in America. Certainly some of the new drugs, we do have some challenges in getting those drugs shipped and available and open on clinical trials here. That's an ongoing sort of issue. A lot in terms of immunotherapies, some of the really early phase trial stuff where they might only be enrolling 10 patients. They'll have very, very specific criteria for patients that will go on to those trials that meet the eligibility criteria, because they're trying to optimize the chance of getting a strong enough signal of success from that trial to justify expanding it into a larger cohort. And so a lot of those studies aren't even open to all the patients. They might be limited centre studies in the United States or Europe, for example. So in terms of a short answer to the question, in general, the therapies that we know are effective are available here in Australia. I'm not aware of a therapy that is so compellingly curative that I would be counselling a parent to get on a plane and go get it. We've explored that. I've had that in the last year or so sadly for a patient of mine, and they just wasn't eligible, even if, you know, they were well enough. That's the other thing, is You know, patients with relapsed AML, you know, unfortunately, it often doesn't hang around in terms of the disease process, and it can be very aggressive and fast, and these patients can be unwell. And so that's an important consideration as well, in that patients need to be well enough to travel. And again, thankfully, most patients don't need to travel because you know, the evidence for those new drugs isn't at that, you know, compelling enough point. We do have, Australia does have a medical treatment overseas program that the federal government fund there for a very, very small subset of therapies where there is a strong evidence base. Invariably, that we had that for CAR T therapy, for example, several years ago, where a panel of Australian experts would get together and consult and agree, yes, here's a case that, you know, the government would support that patient going overseas to access CAR T cell therapy. But again, therapy at the time was pretty compelling, and you've got to have the, you know, the appropriate patient that's likely to benefit from that therapy. And then it wasn't too long, and that therapy was available in Australia, thankfully. So it's all part of the process. But the short answer is the vast majority of drugs are accessible. Some of them can be a bit of a process to access those drugs on compassionate access schemes or other mechanisms. But there are challenges in opening larger studies, sometimes because of drug access and things. And that's an ongoing issue that the Australian and New Zealand Children's Haematology Oncology Group, ANZCHOG, which is the peak professional body for childhood cancer in Australia and New Zealand, which I'm a part of. I'm the vice chair of ANZCHOG. That's an issue that we lobby federal government for and advocate strongly on behalf of all childhood cancer families to make sure that we really minimise any logistical and regulatory barriers to novel drugs finding their way to Australian children.
I know we've touched on transplant, but I guess we can't do a transplant without donors. So are donors easy to find for transplants currently?
Good question. So there's been a number of recent development, or relatively recent developments, in terms of finding donors. So often, as you'll be aware, we look initially at a sibling donor, but you know, you've probably roughly got a 25% chance of being a sibling donor. Unrelated donors, we've generally don't have a lot of difficulty. Again, there's lots of factors in determining whether or not unrelated donors are available. But we also have the option nowadays of using a parent as a half-matched donor. Again, there's some recent developments in how we're able to manipulate what we call the graft, and so that we make sure that we're giving the cells that allow for engraftment without great risk of graft versus host disease or rejection. So, and then there's also, you know, cord blood units. We tend to see sometimes there's challenges in patients of ethnic minorities or in mixed ethnicity families, simply because a lot of the international bone marrow donor registries, the predominant ethnicity would be you know, Western European, Caucasian, and so that can introduce some challenges because of the HLA typing can be influenced by ethnicity. That's where cord blood has been a useful resource because it doesn't have to be perfectly matched. And so that's been useful and remains a reasonable option for a donor source. So but the advent of what we call haploidentical transplantation, where you can use a parent, has expanded the options that we have for transplant in terms of donors. Again, it's a very highly complex area and lots of variables that go into the decision-making process in terms of donor selection, who's the best donor, and you know, when can we get the samples, when can we get the graft and whatnot.
Of course.
You know, COVID had its additional challenges in that space
Sure
as
did.
well. Having to get you know, get the graphs, freeze them in advance, et cetera, et cetera. So
yeah, I think for us it was about a 4 to 6 week turnaround to from the cells being collected to Charlie receiving them.
And I think cord blood came into its own a bit during the COVID because it was more accessible, and that's one of the things I heard, that cord blood was really appreciated. Cord blood donors were really appreciated during that time.
I wish we could donate it here in SA. Unfortunately, that's not something we can do with the cord blood. So yeah. So we're going to wrap that up now. Thank you so much, everybody, for joining us. If you want any more information, you can go to our Facebook page, or you can donate at chust.org/project/Charlie's Rainbow. And the webinar will be recorded and will be available to be viewed on our Facebook page. And you'll be able to ask some more questions and we can get back to them, those that haven't today. So thank you so much for all being here. Thank you, Richard and your team. And thank you, Andy, as well, for sharing your time with us today. That's been brilliant and we really appreciate it.
thank you. Pleasure.
Yeah, thanks, Andy.